Well, Megan and Lauren just gave a great talk showing you microbial are all around the world and they influence the health of Earth, plant, and actually you as well. However, it is very difficult to understand the function of bacterial communities since it typically contains thousands of species, millions of cells, and they talk with each other. The interaction determines function. Just like us, actually, in your lab, you either collaborate with each other or, hopefully not, fight for authorship, equipment, cookies. And all their interactions influence your productivity. So people try so hard to study the bacterial interaction by isolating each individual species and pairwisely mix them together and see how they interact. But there are two challenges. First, we are not able to isolate all the species. Second, even if we have all of them, we need to do millions of experiments to test all the interaction. I can tackle this problem by using a very simple idea, bubble. When you pour a little bath soap into water and stir, you get millions of bubbles. Similarly, I make bacterial solution with oil and surfactant and stir. I get millions of liquid bubbles. The bubbles randomly capture some bacteria together and separate them from other bacteria in other bubbles. This allows us to test millions of interactions in parallel without isolating any single species. This is pretty, but there's still a problem. The sizes are not uniform, which means big bubble has more cell, small bubble has less cell. It causes difficulties on testing pairwise interaction because we really need 2 cells in each bubble. To achieve this, I made a microfluidic chip. This is a small chip with a micro-scale channel in it. Using it, I can generate 8 million uniform-sized bubbles like this in an hour. This is just a start. I also designed another chip to merge the bubble with other reagents. Then I can detect the interaction in each individual bubble in the whole mix. Because the size of the bubble is very, very small. It's just as tiny as diameter of your hair. So all the bubbles is just in a very small tube, instead of using millions of plastic tubes and tons of medium. The small tube can tell us the interaction and also help us to understand the function of bacteria community. Small bubbles can help with answering big questions. I'm Fangchao. I'm a bubble player.